As introduction, basically as you know, tumors also release antigen and can be detected by immune response. This is known as a tumor antigen. The antigen can be released either into the bloodstream or remain on the blood surface. Make it be classified into two types, which are tumor-associated antigen TAA and tumor-specific antigen TSA. Basically, TAA is originated from any protein or glycoprotein synthesized by tumor cell and localized at any subcellular compartment of the tumor cell, such as membrane-bound or nuclear localized. In normal cell, it be expressed at low level and usually high for tumor cell. TSA found only on the cancer cell but cannot be found in normal cell. Mostly, it's derived from oncogenic driver mutation that produce novel peptide sequences.
Immune system serves as the first line of defense against pathogen and cancer. It blocks microbial invasion through a epithelial barrier, disrupt many microbes that do enter the body, and is capable of controlling and even eradicating infection. In tumor, innate immune cells are attracted to the tumor site, where they can recognize the transformed cell. The type of reaction of innate immune system are inflammation and antibody defense. Inflammation consists of the accumulation and activation of leukocyte and plasma protein at the site of infection or tissue injury. These cells and protein act together to kill mainly extracellular micro and to eliminate damaged tissue. Innate immune defense against intracellular virus, even in the absence of inflammation, is mediated by natural killer NK cell, which kill virus-infected cells, and by cytokine called type 1 interferon, which block viral replication within host cell. The innate response to tumor rejection started with a tumor detection induced the activation of the innate immune cell by releasing the damage-associated molecule pattern DAMP and tumor antigen. Then, the application of the innate immune response such as macrophages, neutral killer, and dendritic cell recognize the DAMP by a pattern recognition receptor PRR. Next, dendritic cell, macrophages, and NK act as a first-line effector to the tumor cell. The recognition will eventually activate the microbiome and pro-inflammatory response such as phagocytosis and cytotoxicity. After that, macrophages and neutral killer cell NK contribute to tumor elimination through direct tumor cell killing. In macrophages, the direct cytotoxic function of macrophages requires activation with various cytokines. Upon activation, the macrophages secrete several substances that are directly involved in tumor cell killing, such as tumor necrosis factor (TNF) and nitric oxide (NO), or they actively take out the tumor cell by phagocytosis. Number 5 show that the dendritic cell detect early tumor antigen released by tumor detection through PRR. Dendritic cells are professional antigen-presenting cells that link innate and adaptive immunity and mediate their polarization into effector cells. Dendritic cells can be subdivided into a two main subtypes such as plasmacytoid and myeloid dendritic cells, which specialize in the recognition such as a toll-like receptor. Toll-like receptors are innate immune receptors mainly expressed on dendritic cells, which play an important role in host defense against pathogen by recognizing DAMP. And lastly, the dendritic cell move to lymph node where they activate the educated T cell, trigger an immunological response. The recognition of invading microbe by TLR on dendritic cell induce pro-inflammatory cytokine production and enhance antigen presentation to lymph T cell. And finally, activate antigen-specific adaptive immune response.
Tumor-associated antigens are comprised of short amino acid peptide segments in which T cells recognize them through specific T cell receptors. These receptors exist either as MHC1 or MHC2, which are located on the surface of tumor cells or antigen-presenting cells (APC). The pathways that are used in processing tumor-associated antigens can be further divided into two pathways, which are exogenous and endogenous pathways. In the endogenous pathway, the unfolded intracellular nucleoproteins are tagged with ubiquitin, which will facilitate the recognition with proteasome that will further degrade the proteins into short peptide fragments. It is transported through several pathways in the endoplasmic reticulum and is loaded to the MHC class I receptor, which will form MHC1 peptide complexes. It will then be transported to the tumor cell surface through the Golgi apparatus. The displayed peptides will allow the recognition for CD8 plus T cells, or are also known as cytotoxic T cells. These cells will then release molecules such as granzymes and porphyrin. Porphyrins will catalyze the pore formation on the tumor cell's plasma membrane, where granzymes will then enter the cell and catalyze the breakdown proteins, resulting in cell lysis. Hence, will facilitate the tumor cell degradation and apoptotic cell death. APCs such as dendritic cells may also undergo tumor-associated antigens processing through the endogenous pathways. It will result in both responses from MHC class 1 and class 2, which mediates cell-mediated immune responses as well as the production of specific antibodies. Meanwhile, for the exogenous pathway, APC will directly ingest tumor cells or their antigens, which have been released from damaged tumor cells and will then be degraded in the lysosomal pathway. The antigens will then be degraded to peptides and loaded onto MHC class 2 receptors and form MHC2 peptide complex for the recognition with naive CD4 helper T cells. The activation of naive T cells is catalyzed by the release of cytokines. It will then allow the maturation of the cells into further subtypes such as Th1, Th2, Th17 and Treg cells that function in promoting delayed hypersensitivity, antibody production through B-cell interaction, inflammation or immunosuppression respectively. Hence, it can be said that the recognition of T cells with peptide MHC molecules through the specific T cell receptors will allow the immune system to properly differentiate tumor antigens from self-antigens. The immune system may later either induce deletion of self-recognized T cells or will result in immune tolerance towards the respective tumor antigens. Immune tolerance is linked with the ability of tumor cells in evading the body's immune response. The activation of T cells requires two signals, where signal 1 is from the presentation of tumor antigens on MHC molecules, and signal 2 is from co-stimulatory molecules, such as the binding of CD28 on T cells with B7 molecules on APCs. Usually, tumor cells only stimulate signal 1 alone and inefficiently signal 2, hence inducing immune tolerance. Cross-presentation can be defined as the presentation of tumor antigens which has been taken up by APCs on the MHC molecules of either class 1 or class 2. Whereas, cross-priming is the activation of naive T cells by both signals 1 and 2. Regulatory T cells are recruited in the periphery by tumor cells, which plays a role in suppressing immune responses. In short, the tumor rejection immune response consists of 7 distinct steps, which are tumor antigen release, where its antigen is then captured and presented for the recognition with specific T cells. The antigens are then introduced to T cells through cross-priming, which leads to T cell activation. This will then allow T cells to further circulate in the blood circulation to locate the tumor cells, where it will then infiltrate the tumor cells. T cells then recognize these tumor cells based on their specific antigens, where it will then mediate tumor killing. After the identification of foreign tumor cells, the adaptive immune system generates a response which mounts an army of immune cells designed to target the specific tumor cells. Finally, the adaptive immune system saves immunological memory of the pathogen through the creation of memory cells, which can be quickly called upon to provide protection if the pathogen is encountered again in the future.
The first mechanism the tumor uses to evade the immune system is by the loss of antigen recognition by immune cells. So, this mechanism can happen in two ways. The first one is when the tumor has gained a way to not produce any antigens that will expose itself to the immune system. And the second is when there is mutation in the genes encoding for MHC class 1 molecule that causes it to not be expressed. In the first case, the MHC class 1 molecule is present, but there is no antigen to be expressed. This can happen because of genetic mutations and also deletions in the genes encoding the antigens itself. Or this can also happen when there is selective pressure done by the immune system itself that favors the survival of tumor variants with no antigens or antigens with low immunogenicity. And in the second case, there is a defect in the genes encoding for MHC class 1 molecule, which is an antigen-presenting cell necessary to present the antigen to cytotoxic T cells for elimination. Cytotoxic T cells cannot recognize antigens unless it is first presented by the MHC molecule. The second mechanism the tumor uses to evade the immune system is by secreting anti-tumor immune response suppressors. So these immunosuppressors are actually biologically active compounds released from the tumor, such as cytokines and growth factors. One example of these compounds is TGF-beta cytokine. This cytokine works by inhibiting T cell activation, proliferation, and also differentiation. So other such compounds also exist, such as the interleukin 10, prostaglandin E2, and also cytomykins. Not only T cells, these compounds also target other immune regulators, such as the cancer-associated fibroblasts, tumor-associated macrophages, myeloid-derived suppressor cells, and also dendritic cells.
Next,
For the mechanisms of tumor evasion, it involves tumor-associated macrophages. It may promote tumor growth and invasiveness by altering the tissue microenvironment and also by suppressing the T-cell response. These macrophages will secrete mediators such as interleukin-10 and also prostaglandin E that impair the activations of T-cell and also the effector function. Moreover, tumor-associated macrophages also secrete factors such as transforming growth factor beta that promote angiogenesis or tumor growth. Next is antigenic modulation. It happens when antibody against tumor cell surface antigen can induce endocytosis, and degradations of the antigen results in immune selections of antigen loss variant. Moreover, certain specific tumor cell antigen also have been observed to disappear from the surface of the tumor cells in the presence of serum antibody. However, they will reappear back after the antibody is no longer present.
These are a few of immunological approaches for cancer therapy. Firstly, immune checkpoint inhibitors. Immune checkpoint is a protein on the surface of the cells that function as regulators of immune activation, which means that it maintains immune homeostasis and prevents autoimmunity. These checkpoints, which also present on the cell surface of tumor cells, can lead to problems when they activate and suppress the anti-tumor immune response, which leads to the development of immune checkpoint inhibitors. Usually after tumor antigen recognition, T lymphocyte will infiltrate into the tumor to inhibit and kill it. However, tumor cells can manipulate the approaches to effect the T-cell mediated killing, commonly due to immune inhibition. The immune checkpoint inhibitors is a type of drug that is used to block the proteins. Major immune checkpoints proteins are CTLA-4, PD-1, PD-L1, and LAG-3. Next, T-cell transfer therapy. The main principle of this therapy is where the immune cells, especially T cells, are harvested from patient's blood or from tumor itself, stimulated it to grow and multiply in an in vitro culture system. After sufficient growth, the T cells will be reinfused into the patient, which hopefully will increase the tumor destruction. There are two main types of T cell transfer therapy. Tumor-infiltrating lymphocyte therapy, where the T cells harvested were originated from the tumor, and CAR T cell therapy, where the T cells were isolated from the patient's blood and genetically modified to encode the CAR protein that can trigger T cells to attack the specific cancer. In addition, there is also T cell receptor TCR T cell therapy, where the T cell was isolated from the patient's blood and genetically modified to encode TCR protein. TCR protein triggers T cells to attack the specific cancer.
Third is monoclonal antibody. Monoclonal antibody are immune system proteins created in the lab that are designed to treat cancer by interact with specific target on cancer cells. Like the antibodies that are naturally produced in our own body, this monoclonal antibody recognize the specific target on cancer cell and mark the cancer cell so that they will be better seen and destroyed by the immune system. An example is rituximab. It works by binding to a protein called CD20 on B cells and some types of cancer cells, causing the immune system to kill them. Other monoclonal antibodies bring T cells close to cancer cells, helping the immune cells kill the cancer cells. An example is blinatumomab, which binds to both CD19, a protein that found on the surface of leukemia cells, and CD3, a protein on the surface of T cells. This process helps the T cell get close enough to the leukemia cell to respond to and kill them. Lastly is treatment vaccine. Cancer treatment vaccines are a type of immunotherapy that treat cancer by strengthening the body's natural defense against the cancer. Unlike cancer prevention vaccine, cancer treatment vaccines are designed to be used in people who already have cancer. Treatment vaccine can help the immune system to recognize and destroy the antigens that found on the surface of cancer cells. This leaves the immune system with a memory that helps it fight those antigens in the future. Cancer treatment vaccine may be made in different ways. Some cancer vaccines are personalized. We can look on the figure at the left side. This vaccine means they are made for just one person. This type of vaccine is produced from sample of the person's tumor that are removed during surgery. Besides that, cancer cells also may be made from our own dendritic cells, which are type of immune cells. The figure on the right side shows dendritic cell vaccine stimulate the immune system to respond to an antigen on tumor cells. One dendritic cell vaccine has been approved, which is Furical T, that is used to treat some men with advanced prostate cancer.
In conclusion, tumor antigen is an antigen that has been released by the tumor cell, which consists of tumor-associated antigen and tumor-specific antigen. The antigen release be detected by the immune response as a foreign substances and has own mechanism in order to reject the harmful tumor cell. However, in certain conditions, immune response become weaker and cannot fight against the tumor. Recently, the tumor antigen have become useful and been used as immunological approaches for cancer therapy.